Okay. So it takes a little bit to get everything on the screen, so it probably always will have a bit of a delay. We're going to look at botany. These are illustrative examples on the AP exam. For ECE, however, this is going to be part of your multiple choice. Okay. Remember, I'm going to have a lecture here, but you can look at the PowerPoint. It's on the Google Drive under ECE Botany. And I will also post other assignments on Google Classroom to go with this. So we're looking first at plant morphology. Morphology is referring to body form. And so plants have two organ systems. One is the shoot, the other is the root, just like you have organ systems. And the shoot is their stems, leaves, and flowers. The roots are anything below the ground, subterranean. So we can have tap roots, we can have lateral roots. And connecting these two systems is going to be the vascular tissue. It's going to transport material between the roots and the shoots. And that's your xylem carrying water and minerals, and the phloem that carries sugar. Plants grow their entire lives. All plants are going to grow from their tips. If you were a plant, your body would be the same size that it was as a baby, but your fingertips, your toes, your top of your head would all be much longer, right? And in fact, a kind of a famous question is, if you put the stake in a tree and you come back 50 years, how high will it be? It's not going to move because they're growing from their tips. So that stake is going to be the same place where you left it. Apical meristem is what makes the primary growth. It does the length. It's located at the tips of the roots and the shoots. Whereas lateral meristem does secondary growth. It is going to do the girth. So all plants show primary growth and length, but only some plants show secondary growth. And those are your woody plants. We're going to first start with the root system. So root tip and the root hairs. So we actually have two types of roots that we're going to look at, but all these roots are going to anchor the plant, absorb water and minerals, and some will also store food. So fibrous roots, which is the one at the top, that's going to be a mat of thin roots that spreads out. We're going to find that in monocots, like grass. They're great cover crops because they prevent erosion. The taproot, which is the one right below that, that has one large vertical root. So these produce some small lateral roots, but that large vertical root is really going to hold it deep in the ground. It's going to use deeper sources of water than the fibrous root. And that's also why it's hard to pull out dandelions. And those are in dicots. Now, monocots and dicots are classes of flowering plants, or angiosperms. And then my third picture here with the little bean, all those little white fuzzy things, those are root hairs. They increase the surface area with a very small volume, just like we saw the villi in the small intestine, alveoli in the lungs, capillaries and circulatory system, nephrons in your kidneys. These are really going to help absorb more water, right? So there's your first, your fibrous, your second, the top, and your third, the root hairs. These can also have a symbiotic relationship with fungi, mycorrhizae. So the fungus gets sugar from the plant, but it also is increasing the surface area even more in the roots so that the plants get even more water and minerals. Okay. Primary growth in the roots is at the apical meristem. You notice that there's the root cap in the picture that's on the right. That's just crushed cells. It's going to release the fluid to kind of help soften the soil so the root can grow through it. And then right above that is your apical meristem, where the new growth is happening. There's three other zones. You have your zone of cell division. That's where mitosis is happening. Elongation, the cells are getting longer because their central vacuoles are filling with water. And then the zone of maturation. This is when they're starting to get specialized. And we have three primary meristems. Protoderm makes your epidermis and your endodermis. So those are protective, just like your skin cells are. Ground meristem is going to be making storage tissues. And then the procambium, that's going to be making your xylem and phloem, your vascular tissue. Okay, and we have modified roots. So you can see some here that are prop roots in storage. And our second thing that we're going to look at now is on plant anatomy, the shoot system. So this is aerial or above the ground. We're going to look at the nodes, internodes, and then the types of buds. Okay, and just like roots, you can have some modified shoots. So nodes are typically opposite or alternating, and you can see them here. Okay, petiole that you see in the picture, those are found in dicot leaves. They attach the leaf to the stem. They're really more part of the leaves. And then the internodes, that is the space between those nodes. Okay. Our buds, we have the axillary bud that is at the top. I'm sorry, that's terminal buds at the top. And that is going to create your auxin. Our auxin is the hormone that lets the plant grow toward light and grow taller. So if you want your plant to have more lateral growth, the axillary buds forming lateral shoots, you would have to take that terminal bud off. Primary growth in our shoots, we have our apical meristem in 1 and 7. Right, so those are again going to make the protoderm, procambium, and ground meristem. Auxiliary bud meristem, that's at the base of the leaf primordia. And then the leaf primordia, which is 2 and 6 here, those are going to give rise to your leaves. All right, last part of our shoot system, the leaves. We'll look at the mesophyll tissue, the veins, and the vascular bundles. So leaves are for photosynthesis. They have chloroplast in them, and chloroplast has chlorophyll and some other accessory pigments, and that absorbs the light energy, and in the light reactions, it's going to turn that light energy into ATP and NADPH. And then that energy is used in the Calvin cycle to fix carbon dioxide as glyceraldehyde 3-phosphate. We're going to have gas exchange. Those are at the stoma or stomata. And you can see them at the picture on the top right. Those are the openings, usually on the underside of leaves. And on either side, those purple cells, those are the guard cells. Those control the opening and the closing of the stomata. And we control that, or the plants control that, because of transpiration. Evapotranspiration, the water will leave through the stomata, and that's how you get, for most plants, from root up to shoot, the water using the xylem. The plants that don't have xylem, like moss, or most moss, they can't do transpirational pull, and that's part of the reason they're so small. These are some examples of modified leaves. Petals are a great example of an adaptation. So plants have these colored leaves to attract pollinators so that they can guarantee that their genes go to the same plant as best as possible, and that increases their Darwinian fitness. So you can see in this picture, we have our cuticle. That's a waxy coating that prevents water loss. Here's our epidermis. Here's our lower epidermis. Those are your skin cells. Those were made by the protoderm. And then here, your palisade mesophyll has chloroplast, as does the spongy mesophyll. Spongy mesophyll is called that because it has these air pockets. Right? Most of your photosynthesis is happening here in the palisade, and that's ground tissue. Then we also have our vascular bundles. Xylem's here on the top, that's carrying water and minerals. Phloem is on the bottom, it's carrying sugar. We'll go over why they're next to each other in a bit. And the stoma or stome is at the bottom. That's just a hole, but it's controlled by these cells on either side called the guard cells. In this picture, you can really see those guard cells. Okay, so the next one, you can see some more of the same things, the leaf anatomy, right? So the epidermal, the ground tissue, and then our vascular tissue. A couple quick reminders. Light reactions we already talked about, they occur in the mesophyll, the chloroplast thylakoids, because that's where the Photosystems are embedded with chlorophyll to capture that light energy. The Calvin cycle in C3 in cam plants occurs in the mesophyll cell still, but in the stroma. And then in C4 plants, because they're dealing with photorespiration, remember that's when oxygen is picked up by rubisco instead of carbon dioxide. And so instead of being a reducing reactions to make G3P, it's an oxidizing reaction that just uses up the ATP and NADPH. So C4 plants instead are going to have their Calvin cycle occur in the bundle sheath. So actually, we went through how the carbon dioxide gets shuffled in there. Okay, and so you have a higher concentration of carbon dioxide there than oxygen. So the rubisco enzyme will only pick up CO2. Most enzymes, remember, are substrate specific, but rubisco evolved when oxygen wasn't on Earth, really. So it wasn't an issue for it. Both systems are going to depend on each other. Roots are going to depend on the sugars produced by photosynthetic leaves. Shoots are going to depend on water and minerals absorbed from the roots. Okay. Now, sugars go in both directions. In this time of the year, sugars are going from the roots up to the shoots to feed the new growth. And that's why we tap maple trees, to get that phloem so we can make maple syrup. Water and minerals only go in one direction, though. They go from roots to shoots. Okay. So at this time, sugar's going roots to shoots also, but as I go into the autumn, it's actually going the opposite direction. The sugars are going from the leaves back down to the roots to be stored so that they'll be available for spring and new growth. All right, plant tissues. You have three types of tissues. Dermal, we said protects the plant. Ground, that's most of your plant tissue. It's going to do the photosynthesis with the mesophyll, but also storage tissue. And then vascular, the xylem and the phloem. And they have three main cell types. Just like you have cell types, if you remember the histology lab, they have cell types. They have parenchyma. This is the least specialized. And that's your mesophyll again in your storage cells. And tissues and leaves, stems, etc. Parenchyma has a kind of uneven, thickened primary cell wall that gives a little bit more structure. And we're going to find that in new growth. And that's the stringy stuff when you eat celery that you get with the celery. Sclerenchyma has a very thick secondary cell wall. And it does support, and it's very rigid, so it's not going to elongate. And it's usually, well, it's always dead at functional maturity. Okay. So here's your parenchyma. It's unspecialized, thin, and flexible. So it's that ground tissue that's being pointed to right here. You can see here's your ground tissue, and all this purple stuff is starch being stored in it. Parenchyma. Here we have, you can see the difference between the parenchyma and the parenchyma. You have a thicker primary cell wall. Primary is just the first cell wall that the plant makes, right? And again, it's going to support without restraining growth. It is still alive. And here's sclerenchyma. It has lignin or wood in it, right? They cannot elongate. Oops, it went ahead on me. Sorry about that. And they're dead at maturity. So there's cells for support. We're going to have the xylem, vessel elements and tracheids, fibers that we use to make ropes, and nutshells, seed coats, and that grittiness in pears, why some people don't like to eat pears. Those are all sclerenchyma. Our vascular tissue, it starts with your ferns. So remember, for mosses at this level, we do not consider mosses or bryophytes to have transport systems. Bryophytes is just a technical term for mosses, right? Xylem is for the water. It's going to use capillary action. That's due to hydrogen bonds between water molecules with adhesion and between the water molecule and the xylem for adhesion, and transpirational pull. Phloem is going to do a mass or bulk flow of sugar. So here you can see the vessel elements. They're stacked together like pipes and they're hollow. Right, here's the tracheids. These are a little bit more tapered at the end, so you can see tracheids, T tapered, and that has a little bit of lateral movement because of that. Right. So water and minerals, they're only always moving roots up to the leaves, one direction. These are dead at functional maturity, and they're basically like piping in the plant, okay? How the tissue carries the water, you have root pressure, that's positive pressure. So as the minerals are transported into the root hairs, water is going to foul, forcing it up, more water, but it's not going to get very far. So positive pressure is always going to increase your water potential. So it's a push. So water's going to want to leave that area of positive pressure. Transpiration is a negative pressure. It's going to be a pull of the water. And that's always going to decrease your water potential. So water's going to want to go where there's more transpiration, so it's going to want to go up those leaves. All right. So positive increases water potential, negative decreases water potential. Solutes, remember, always decrease water potential because our solute potential formula is negative I CRT, right? So it's always a negative number. And so water is always going to want to go where there's more solutes and where there's more negative pressure, and it's going to want to leave an area where there's positive pressure. Right? This transpiration is going to be controlled by the stomata, those openings, or sometimes they're called stoma. It's in both ways. So what happens is blue violet light induces pumps to work in the guard cells and potassium ions to go into these guard cells. So all these potassium ions are going into these guard cells. Water potential now just decreased, so water's going to follow in there. And these get turbid, they're filled with water, and that causes them to pull apart, kind of like balloons. If you have two balloons and they're just kind of here, and then you blew them up, they would pull apart like that, right? And then in order to close them, the potassium leaves and the water leaves. So blue violet light, that's what causes that pump to work. Other things, decrease in carbon dioxide in the airspace, that will cause them to open, and internal clocks. We'll also see that there's a hormone that helps control this as well, abscisic acid. We'll talk about that more later in the slideshow. So how does water get from the roots to the xylem? Well, there's two ways it can go. Symplastic route, it travels through the cell cytoplasma, using the plasmodesmata. Remember, those are gaps in the cell wall back to the cell unit. And so they have to pass through that plasmodesmata, through the phospholipid bilayer. It is getting, if you filter, basically, checked. The apoplastic route, it travels through the cell wall, and it doesn't allow it to get checked. But there is, before it can enter the steel, there's what we call a casprin strip. It's wax. So remember, wax, hydrophobic, it's a fat, and that kind of forces any water going the apoplastic route to have to pass through at least one membrane and get filtered, because you don't want anything, it'd be like allowing anything into your own circulatory system. If it's something bad, you don't want to allow it in. Same thing for a plant. Okay, so that's that casprin strip that we see here. And the steel is where the vascular tissue is, the xylem and the flow. Right, so we're seeing regulation again. Phloem is your food conducting cells. So these go in both directions depending on this season. And you have this companion cell here, and this is your sieve tube. Now the sieve tube is hollow, but it's alive. And the reason it's alive is because of this companion cell. The companion cell has a nucleus, it does protein synthesis, it makes energy, and it's basically kind of like, it's just keeping the sieve tube alive, okay? It's like life support for the sieve tube. And then we have, again, you can see the sieve plate here has all those holes, those plasmodesmata for the sugar to go through. All right, and here's another picture of that. Those sieve tube elements and companion cells make up phloem. So these are living unlike xylem at functional maturity. They control, they do, not the companion cell, but the sieve tube loses its nucleus, its ribosomes, spectral. Okay. And the sieve plates, those are those pores to facilitate the flow of fluid between cells. And companion keeps them alive. All right, phloem is going to carry the process of photosynthesis from source to sink by translocation. We've seen the term translocation used in a lot of different references. You just always have to think about what are they talking about, what overall subject. So here we're moving something, translocating, and we're going to do it in the phloem. So the sieve tube members, that's where the sugar is going to be. The companion cells are going to keep those sieve tube members alive, but they also help load the sugar. Right? So how this is done, this is an example of active transport using a co-transporter. So hydrogen ions are pumped out using the energy from ATP here. Right? You get an electrical chemical gradient, lots of potential energy. And the only way the hydrogen ions can go back down both their concentration and their charge gradient is through this co-transporter. When they do and they release that potential energy, it's used to transport sucrose into the phloem. When that does, I've got an increase of solutes. And remember what increase of solutes do? It decreases my water potential. Water wants to move in there, and that's literally what happens. This is why xylem, if you remember in the veins, is right next to phloem. So the water enters in, right? That causes a pressure buildup, and that's what's going to really help force the sugar from source to sink. When it gets to sink, it gets put to storage or it gets used by leaves for growth, depending on the season. The water enters back into the xylem. Okay? If I'm looking at a dicot, I'm going to find, if I look at their stems, here we have the phloem on the outside. This is your vascular cambium, and there's your xylem. It's in a ring all the way around. And if I look here, they're kind of spread throughout. Okay. If I look in the vascular tissue in the roots, in a dicot, you're looking for this kind of X, right? That's your xylem. The stele, again, not just stele is where the vascular tissue is, but, oops, this goes a little too fast there. If I look here at my monocot, you see in the root, and it's very, you have to be careful, because in the root of a monocot, you see that here's your xylem in a ring, here's your phloem in a ring. But in the stem, if I see it in a ring, it is a dicot. So you have to be careful there. All right, so regions of growth, those are the meristems, and they grow their whole lives. We talked about the apical meristem, primary growth growing from the tip. All right, and then lateral meristem, secondary growth. Not all plants show that. That's growing in girth. All right, so here's our apical meristem again. And that's our primary growth. This first part that comes up is called the coleopedal. And you can see here is your axillary buds. That's going to allow for the lateral growth. They're right now not growing because up here in the apical meristem, there's a hormone auxin that is preventing them from growing out because it's trying to grow up first so that it can get more sunlight. All right. Here in a woody plant, we have primary growth still with apical meristem, but we also have secondary growth. And that's your lateral meristem. And we have two types. You have the vascular cambium. That's what we all see here between the phloem and the xylem. So it's going to be making new phloem and new xylem. The old xylem is getting filled up with sap and stuff, and that's the wood. And in fact, on your tree, the center of your tree is completely dead. It is this outer part of the tree that's alive. The old bark here, the old phloem, that usually comes off. You'll see it on some trees. Maple trees, for instance, you can often find it at the bottom. This is why, like, a carpenter ant can eat the whole inside of your tree and the tree will be fine. Or if you look at the pictures or been out and seen the redwoods, they can put a tunnel through them and the tree will be fine. Okay, but if you removed all the bark, you girdled the tree, it would kill it. Right? So vascular cambia makes your secondary phloem and secondary xylem. And then you also have cork cambium that is going to make cork. Right? So our secondary growth is the diameter growth. So here you can see you have your phloem and your xylem. This is, and you have your vascular cambium. Here's our bark. And this is each year's growth. So you might remember you've counted probably rings on trees. That's how you can tell how old it is. The wider xylem that I see here, that's called springwood, and that's because there's more water in the spring. So the cells get larger. And then here is your summerwood, and you can see the cells are getting smaller, and that's because there's less water, and that's what creates those rings. Okay. So here's your cork, here's your vascular cambium. This side, phloem, this side, xylem. Okay, and then everything below that, that's last year's xylem. So again, forming those rings so you can tell how old. And the old phloem, the old bark, that comes off at the tree. It just sloughs off like old skin, I guess. Okay. And this tree, how old is it? Just count your rings, right? 1, 2, 3. 3 years old. And this is girdling a tree. That's where you remove that bark. So you're removing the cork cambium, the phloem, the vascular cambium, and that's going to kill it. Where will the carving be 50 years? Same spot. Doesn't move. All right, plant hormones regulation. These I would expect could be on the AP exam. So we have auxin, gibberellins, abscisic acid, ethylene, and there's a lot more hormones, but these are the 4 we're going to really focus on. There are ones that are steroid-based for them. They have florigen, which is a hormone that allows them to flower. But we're just going to focus on these 4. But if you go on and take a botany course, which you might have to do, because I know botany's on the dental exam and the medical exam. So if you want to go into those fields, you would take one. But we're just going to focus on these 4. So if you took a botany course, they would do a lot more. Auxin, sometimes they'll write as IAA. This controls cell division and differentiation and phototropism. Now, this was studied by Darwin, and what they saw was the auxin builds up here at the shady side of the coloidal, that early stem, and these cells are going to elongate, and that causes it to grow toward the light. It also does apical dominance. So it's made in the terminal bud, and it prevents the lateral buds, as we talked about before, from growing out, right? So the plant's going to grow first toward the sun. Right? And you can see these axillary buds here, they're being prevented from growing out because of that auxin. But if I take off the auxins being made, you can see they have started to branch out laterally. So how auxin elongates. So increase auxin away from the light, proton pump gets activated, hydrogen ions are transported into the cell wall, that lowers the pH. Remember, different enzymes have different optimal pHs. These ones work at a low pH. That breaks the cellulose cross-links in the cell wall. Water fills the central vacuole, and now the cells get longer on the shaded sides. That's what causes it to bend toward the light. Gibberellins, these are a whole group. There's over 100 different types. And they have several effects. One is stem elongation. they actually do something called bolting. It's called foolish rice disease. It was discovered in the 1920s in Japan. And they called it foolish rice because the rice plants would go really, really tall suddenly. And they were so tall that they couldn't support their own weight. And they'd fall into the rice paddies and rot. And when they studied it, they found out it was caused by gibberellins. Fruit growth, and then seed germination. So sometimes that's why you see these, if you ever see like natural grapes, they're usually not as large as these grapes that you see in the supermarket. And that's just treating them with different hormones. Seed germination, they're really important because seeds won't germinate. If you've ever planted seeds, the first thing you do is you water them. The reason you do that is to get rid of another hormone that we'll talk about, but it also helps activate the gibberellins. And the gibberellins causes an enzyme called alpha amylase. You might remember amylase when we did digestive system, salivary amylase and pancreatic amylase, breaks down carbohydrates. Here it's breaking down the endosperm, the carbohydrates that are here, so that the embryo can have that energy to start to grow. Okay. Abscisic acid slows growth. It is actually antagonistic to gibberellins. This causes seed dormancy, and this is the one that gets washed away while you start to water your seeds. And it really is important because it only allows seeds to germinate under optimal conditions, light, temperature, and moisture. Ethylene, this is a gas hormone, and it is involved in fruit ripening. So that's one of the reasons you never store your onions near any fruit or anything else, because they actually produce a lot of ethylene and it'll cause your food to go rotten much quicker. Also, leaves dropping in the fall. Okay, and we saw apoptosis. And if you were here when you were around 2011, or 2010, I can't remember, Storm Alfred, you'd be in about second grade, most of you. We know now why leaves dropping, apoptosis, is so important, because without it, all that snow weighed down those trees and they started to break. Caused a lot of problems, killed a lot of trees. You have this in your book, all right? Kind of nicely goes over all of them. If you're taking the SAT 2, you would have to memorize them. It's a spit back test. Fruit ripening itself is an adaptation. Right? Hard, tart fruit, that protects the developing seeds from herbivores. Ripe, sweet fruit attracts the animals. So the seeds have matured. All right. And then the orangutan, for instance, here, eats this and walks or travels and then defecates. And the seeds will come out, they're protected by the seed coat, and it's caused the dispersal of the seeds away from the parent plant, so it has a better chance of growing. All right. So what happens, we're going to try a ripening process. You break down the cell wall so it softens. You convert the starch to sugar. That makes it sweet. And ethylene is positive. Once you start that production, you will have more fruit ripen. Okay. Apoptosis, that's the response to ethylene and auxin, so death of annual flowers, differentiation of xylem, that loss of cytoplasm, and the shedding of autumn leaves. Tropism. So when we were doing animal behavior, we talked about taxis, which is a directed movement toward a stimulus, or kinesis, which is a random movement until you hit the stimulus. Plants can't move, but they can grow away or toward a stimulus, and that's what tropism is. Positive is toward the stimulus, negative is away. So phototropism, photo, light. So that's due to auxin. Stems show positive, roots show negative, right? Thigmotropism, that's to touch. So you can think of a pea plant growing up a trellis would be a good example. Gravitropism is moving toward or away from gravity. So it's negative in shoots and it's positive in roots. And we think it's because of these like starch molecules. But there was a cool installation where they did these upside down, and then the roots, because they were facing this way, started to curve like that, and the shoots started to curve down like this, and it was really neat looking, but I can't find a picture of it. Hydrotrophism, that's the movement toward water. This is a big problem in West Hartford because the trees like water, and in a lot of the yards, if you go look outside, you'll see there's a tree in the front yard. They put them right near the sewage drain, which has water. And so the tree roots want to find the water, they'll grow toward it, and then they can break the sewage pipes, which is very expensive. Turgor movements, rapid and reversible response to turgor pressure. So it's putting water into the cells or removing the water out of the cells quite rapidly or quickly. So here you have your Venus flytrap, and it closes to eat this insect. It isn't eating it because it needs food. It's eating it because, like all of these plants, it doesn't have enough nitrogen in the soil. Same thing with pitcher plants as well. Or it can be to avoid herbivores. We talked before that we have plants that if you brush against them, their leaves will fold, so they look unappetizing to your herbivores. And then nastic movement, this is the migration of potassium. At night, it causes the leaves to fold up like morning glories, right? They don't need to be open at night. There's no pollinators for them at that time. We had talked about photoperiods when we were doing animal behavior as just an example of other organisms besides animals respond to their environment. But I just wanted to quickly go over this. So here it's light that we initially thought caused them to flower not. But it's actually not the light, it's the night. They have a critical night. So long day plants need a short night, and short day plants need a long night. And if I interfere with a short day plant's night with a flush, it will not flower like this one, okay? And this has been a problem with global climate change because we're changing temperatures, we're changing a number of different things, and some plants are not flowering. All right, that is your first lecture on botany. So make sure you have filled in the notes. If you have not filled in the notes, go back, fill them in, and I will put up the second one as well. And this is, again, it's on the Google Drive too.